Good evening, students. Today we are having our day 35 and it's the next video in sequence for class 12th. Today we will study about megasporogenesis. In our previous video we studied about types of ovule. Now we are having the process megasporogenesis, which will help us to form megaspore, or we can say we are going to develop a female embryo sac or the female gametophyte. Fine. So in case of megasporogenesis, what is this process? The process in which megaspores are formed from megaspore mother cell through a single meiotic division. Here also a single megaspore undergoing meiotic division will form 4 megaspores, or we can say megaspore tetrad. So let's go. The nucellus. We studied about nucellus. It's a mass of nutritive material which is present inside megasporangium. Megasporangium is what? Our ovule. Ovule के पास nucellus present होता है जो mass है nutritive tissue का. From that nucellus, one of its cell which is present towards the micropylar end will behave like archaeosporium, or we call it archaeosporium cell. This is single celled in nature and hypodermal in origin. Just below the epidermis it is present. So this is the cell of one of the cell of nucellus which is present in micropylar end. Fine. This archaeosporial cell, it is diploid in nature and it will undergo single mitotic division and form two cells which are also diploid in nature, because in case of mitotic division the chromosome number remains same. So here single that one archaeosporial cell, it will undergo mitotic division to form primary parietal cell, and the other one is the primary sporogenous cell. This primary sporogenous cell will behave like megaspore mother cell and it will undergo single meiotic division to form 4 megaspores, or we can say megaspore tetrad in linear arrangement. In that linear arrangement of megaspores, the chalazal one, the upper one, the chalazal one megaspore remain functional and it will enter the procedure for the formation of female gametophyte, or we can say female mature embryo sac. And rest of the 3, the micropylar ends megaspores get degenerated. There is a twist in this case. What happens? We studied about types of ovule on the basis of nucellus, whether they are developed or not developed. In case of developed ovule, what we call them? Crassinucleate ovule. So in case of crassinucleate ovule, what happens? This process is followed in this manner, where the archaeosporial cell will undergo mitotic division to form two cells. One is the primary parietal cell and the other is the primary sporogenous cell. And this primary sporogenous cell will behave like megaspore mother cell, and being diploid, it will undergo meiotic division. One meiotic division which will give rise to the formation of 4 megaspores, which are linear arrangement of megaspore tetrad, and out of these linear arrangement only one megaspore is functional, and that one megaspore will be the chalazal one, and rest of the 3 micropylar end megaspores will get degenerated. Fine. So here one more thing is there. In tenunucleate ovule, it's a kind of ovule in which the nucellus is poorly developed. In that case, this archaeosporial cell itself behave like megaspore mother cell and it will undergo the meiotic division to form 4 megaspore. In that case also only one megaspore is functional. Okay? So this is about the megasporogenesis in angiosperms. We considered 2 different examples, one in case of crassinucleate ovule and the other one in the tenunucleate ovule. The procedures are totally same. What happens? Megaspore mother cell will undergo only one meiotic division to form 4 megaspores, but the cells are different, which will behave like megaspore mother cell. As in crassinucleate, this archaeosporial cell will undergo mitotic division to form parietal cell and sporogenous cell. So here in case of crassinucleate ovule, sporogenous cell will behave like megaspore mother cell and it will undergo meiotic division to form 4 megaspores. Whereas in case of tenunucleate ovule, what happens? this archaeosporium itself act as megaspore mother cell, and this cell will undergo the meiotic division. It will undergo the meiotic division directly to form 4 megaspores. But in both the cases, only one megaspore is functional. That is the chalazal and one megaspore. Right? Let's come to the next topic here. Embryo sac. The female gametophyte or the mega gametophyte can be of multiple types. So according to P. Maheshwari, they divided embryo sac in 3 categories. One is the monosporic, bisporic and tetrasporic. This embryo sac is divided into these 3 types on the basis how many megaspores will convert into a mature embryo sac. Monosporic development, where single megaspore will develop into mature embryo sac. This is known as monosporic development of embryo sac. It is observed in Polygonum and Enothera. Bisporic development, where 2 megaspore will develop into mature embryo sac. In that case, example is Allium cepa. After that, tetrasporic where all the 4 megaspores will mature and develop into their individual mature embryo sac, and it is observed in Rusa and Plumbago. Fine. So this is all about what is megasporogenesis. Secondly, we studied about how megaspores are formed in case of crassinucleate ovule and in tenonucleate ovule, which cells are mainly referred as megaspore mother cell. And in third part, we studied about the embryo sac, how many types of embryo sac may be there in any plant, but in majority, monosporic development is observed. And it was given by P. Maheshwari. Okay, in my next video, we will study about the complete development of female gametophyte, how it will become 7 cells and 8 nucleate structure. Thank you.